All right, let's dive into one of the most incredible processes in all of biology, how a cell moves things around. We're talking about how it gets food in and takes the trash out. It might sound basic, but this constant, perfectly controlled traffic is literally what makes life possible. So let's start with a big question. How does a cell, this tiny little building block of life, actually stay alive? How does it get what it needs, get rid of what it doesn't, and talk to its neighbors, all while protecting itself from the outside world? The answer is just so cool and elegant. It survives by being an expert border patrol agent. You have to think of the cell membrane not as a static wall, but as this incredibly smart, selective gatekeeper. It's constantly making decisions about what gets a VIP pass and what gets turned away at the door. Okay, so let's zoom in on that smart border, the membrane itself. To really get what makes it so brilliant, we need to look at the different ways it manages all this traffic. And we're going to start with the easiest method of all. And that's called passive transport. The key word here is passive. It means the cell doesn't have to spend a single drop of energy. Seriously, it's like floating down a river. You're just going with the natural flow of things. So the whole idea behind passive transport comes down to something called the concentration gradient. That's just a fancy way of saying things naturally move from a place where they're super crowded to a place where there's more space. And as you can see here, there are a couple of main ways this happens: simple diffusion and facilitated diffusion. First up, we've got simple diffusion. This is like the express lane for really small, uncharged molecules. Think oxygen and carbon dioxide. They're so tiny and neutral that they can literally just slip right through the oily membrane. No questions asked, no help needed. This diagram shows it perfectly. Just imagine you drop some food coloring into a glass of water. At first, it's all clumped together in one spot. That's area A. But then the molecules just naturally spread out into area B until they're evenly mixed. That right there? That's diffusion. It just happens. Okay, but that leads to a pretty obvious problem, right? What about things the cell desperately needs that are bigger or have an electrical charge, like glucose? Sugar, the cell's main fuel source. It can't just sneak through the membrane. So how does it get inside? Well, it uses facilitated diffusion. The word facilitated just means helped. The cell has these specialized proteins embedded in its membrane that act like bouncers or special doorways. They create a specific path for molecules like glucose to get across. And here's the kicker: it still doesn't cost the cell any energy because it's still just moving from high concentration to low. A really great example of this is the GLUT1 transporter protein. Think of it like a custom-made revolving door that only fits glucose. It sees a glucose molecule, grabs onto it, and then it physically changes its own shape to shuttle that molecule across the membrane and into the cell. It's absolutely essential for feeding our cells, especially our brain. And the whole process is like this elegant little four-step dance. First, the glucose binds to the protein from the outside. That connection makes the protein change its shape, so it opens up to the inside of the cell. Then, the glucose is released, and finally, the protein just snaps right back to its original shape, ready for the next one. So cool. All right, let's pause and look at this comparison. On the left, you've got passive transport, which we've just covered. See that energy row? No energy required. But look over on the right, there's a whole other category: active transport. And the big difference is right there. It demands energy in the form of ATP. And that brings us to our next major topic: active transport. If passive transport was just floating down the river, active transport is like grabbing a paddle and fighting your way upstream, completely against the current. So this is the most important thing to remember. Active transport moves things against their concentration gradient. It's forcing molecules from an area where there aren't many to an area where they're already crowded. And to pull that off, to push something uphill like that, the cell has to spend its energy currency, a molecule you might have heard of called ATP. The most famous example of this is the sodium-potassium pump. This little machine is a workhorse running nonstop in almost every single one of your cells. It's the reason your nerves can fire and your muscles can move. For every bit of ATP energy it uses, it shoves 3 sodium ions out of the cell and drags 2 potassium ions in, both of them completely against their will, against the direction they'd naturally flow. And get this one process is so unbelievably critical for our survival that it can use up to a quarter of a cell's entire energy budget. That's a huge amount of power for just one tiny molecular machine. It really tells you how important this is. Now, the cell is also super clever. It's not going to waste energy if it doesn't have to. So it uses something called co-transport. Basically, it uses the energy that it already spent creating a gradient, like that buildup of sodium outside the cell, to give another molecule a free ride inside, even if that second molecule is also moving uphill against its own gradient. And this comes in two main flavors. With symport, both molecules move in the same direction. It's like one molecule is rushing downhill and it just grabs the other one and pulls it along for the ride. With antiport, they move in opposite directions, kind of like a revolving door where a molecule leaving powers another one coming in. But what about the really big stuff? I'm talking about things like bacteria or huge protein molecules. They're way too big to fit through any kind of pump or channel. For these massive packages, the cell has to use a totally different strategy called endocytosis and exocytosis. So let's check out phagocytosis, which literally translates to cell eating. It's amazing. As you can see in the diagram, the cell's membrane actually reaches out, surrounds a big particle like a bacterium, and completely swallows it, pinching off to form a little bubble inside itself called a vesicle. Then it can break it down and digest it. And this slide, this is just awesome. On the left, you see that neat, clean little diagram. But on the right, that's the real deal. Those are actual microscope images of a cell in the middle of eating something. You can literally see the membrane wrapping around its target. It's not just a drawing. It's this dramatic, powerful event happening inside us all the time. So let's step back and bring this all together. All of these different transport methods, the passive, the active, the bulk shipping, they're all happening at once all the time, just to maintain a very specific, very stable environment inside the cell. It's all about maintaining a vital balance. So what are the big things to take away from all this? Well, remember that passive transport just goes with the flow. No energy needed. Active transport fights against the current, and that costs energy. That costs ATP. Then you've got bulk transport for the really big stuff, using those vesicles. And all of it working together maintains that delicate internal balance that is absolutely essential for life. This constant buzzing traffic is the very definition of being alive. But it leaves us with one last, really important question to think about. What happens when this perfectly tuned system gets broken? What are the consequences when the gates get stuck open or the pumps suddenly fail? Well, that's where we find the root causes of so many diseases.